Imagine this for a second. What if the key to unlocking a cure for one of the most stubborn, like completely drug-resistant cancers on the planet wasn't, I don't know, some futuristic billion-dollar synthetic chemical?
Right.
What if instead it was just hiding in plain sight in a really common decades-old treatment for malaria, just, sitting in pharmacies worldwide waiting for us to realize what it can actually do?
It sounds almost like a thriller premise, right?
Yeah.
But it is the exact reality of this huge breakthrough we're looking at today. It's from a brand new July 2026 paper in Cell Death and Disease. And it just completely upends how we think about treating colorectal cancer liver metastases.
Yeah, and colorectal cancer liver metastases, or CRCLM, is an incredibly grim diagnosis for you to get because colorectal cancer is already, one of the most prevalent malignancies globally.
Absolutely.
And when it advances, the cancer cells frequently migrate through the bloodstream and they basically seed themselves in the liver. And once it metastasizes there, mortality rates just skyrocket because liver tumors are, well, they're notoriously difficult to treat.
They are. But, modern oncology isn't completely without its weapons here. The current cutting-edge protocol is this combination of two really powerful drugs.
Right.
First, you have Regorafenib. This is a multi-kinase inhibitor. So its job is to block specific enzymes to stop the cancer cells from dividing. But crucially, it acts on the tumor's infrastructure.
The blood supply, right?
Exactly. Tumors build these really chaotic, leaky blood vessels to feed themselves, and Regorafenib normalizes those blood vessels, which, in theory, at least helps other drugs actually penetrate the tumor tissue.
So Regorafenib is trying to stabilize the supply line so the second drug can get in. And that second drug is an anti-PD-1 monoclonal antibody, which is an immunotherapy. The entire premise of PD-1 inhibitors is to just take the brakes off the patient's own immune system, specifically waking up the CD8 plus T cells so they can, recognize and destroy the cancer.
On paper, it is a brilliant two-pronged attack. You normalize the blood supply and hit the tumor with this massive immune response. But the conflict, and really the entire reason this new research is so pivotal, is that in real-world clinical practice, a staggering portion of patients are non-responders.
Yeah, that's the tragedy of it.
They receive this heavy-hitting combination therapy, and the tumor it simply ignores it.
The combination completely fails. And to understand why it fails and how this wild malaria drug actually comes into play, we have to look at the underlying genetics.
Right, the root cause.
Yeah, because the non-responders almost universally share a very specific villain, which is a mutation in the KRAS gene.
The KRAS mutation is present in about 50% of all colorectal cancer patients. And the researchers in this study, they focused on the most common variants, specifically KRAS, G12D, G12V, and G13D.
Okay, so let's unpack this for a second.
Right.
To understand what that mutation means, you really have to look at what normal KRAS does. If you picture a healthy cell as a car, the normal KRAS protein is essentially the gas pedal that tells the cell to grow.
That's a good way to look at it.
But it's a tightly regulated pedal. It only presses down when it receives a specific signal, and then it immediately lifts off. But when it mutates, when you get that G12D variant, for instance, it's like someone dropped a brick on the gas pedal.
Exactly. It's no longer a regulated gateway. It's a hot-wired system where the doors are permanently stuck open, and it's just flooding the cell with this constant, never-ending command to grow and multiply.
And that mutation, that single broken switch is why, for decades, oncology has referred to mutant KRAS as completely undruggable.
Yeah, undruggable is the famous label for it.
I really want to dig into that word, though. Why can't we just design a small molecule inhibitor to bind to the broken switch and turn it off? We do that for dozens of other cancer mutations, right?
We do, but it comes down to the physical three-dimensional shape of the protein itself. Most targeted therapies require a binding pocket. They act like a key sliding into a very specific lock, or they need a deep groove on the protein surface to latch onto securely.
Right.
But the surface of the mutant KRAS protein is notoriously shallow. It's incredibly smooth.
So it's like trying to get a rock-climbing grip on a greased bowling ball.
That is exactly it. There's just nowhere for a traditional drug molecule to find any purchase. It just slides right off.
Wow. Okay, so because small molecule drugs just slide off, the mutant KRAS is left totally free to initiate this massive signaling cascade.
Right. It probably switches on two primary downstream pathways. Specifically, these kinases known as pERK12 and pACT. These are basically the internal wires transmitting that constant grow command right into the nucleus of the cell.
Okay, so let's do a quick recap here. The standard Regorafenib and anti-PD-1 combo fails in so many patients because of this smooth, hyperactive mutant protein called KRAS, which basically keeps growth signals. The PERK12 and PAC just stuck in the on position. But wait, I'm stuck on something here. I want to throw this question out to you, the listener, as well. If the tumor cell is growing out of control, driven by this blatantly mutated protein, why doesn't the anti-PD-1 immunotherapy just tell the immune system to attack it?
That is the big question.
I mean, immunotherapy relies on T cells circulating in the blood. If a T cell bumps into a tumor cell, it should recognize it as abnormal and attack it, regardless of how fast it's growing, right? How is an internal genetic mutation stopping our own external immune system?
And that is the core mystery this paper unravels. Mutant KRAS doesn't just drive growth. It uses those same downstream pathways, the PERK12 and PAC we just mentioned, to actively build a fortress against the immune system.
How does it do that?
Well, when those pathways are permanently on, they do something incredibly devious. They heavily upregulate a transcription factor called MYC.
MYC. And a transcription factor being, a protein that binds to DNA to turn other genes on or off.
Right, and MYC acts as a master silencer for a critical defense mechanism in the body called the interferon response, or IFN response.
Okay, interferon response.
Yeah, in cellular biology, the interferon response is essentially the 9/11 system. If a normal cell becomes infected with a virus or starts turning cancerous, it triggers the IFN response to broadcast a distress signal. But MYC completely crushes that system.
Oh, wow. So if the cellular 9/11 system goes down, what exactly stops happening, like at the cellular level?
Two critical things disappear.
Okay.
First, the tumor stops producing a chemokine called CXCL10.
Okay.
Chemokines are chemical attractants. So in a normal immune response, CXCL10 acts like a chemical flare. It creates this breadcrumb trail that guides T cells directly to the site of the problem.
Got it. So without the CXCL10, the T cells are essentially blind. They just circulate right past the tumor without even knowing it's there.
Exactly. The flares are extinguished. But even if a T cell accidentally bumped into the tumor, it wouldn't know what to do because of the second thing MYC shuts down.
Right. There's another layer to the invisibility.
Yes. The second thing is a protein complex called HLA-ABC. You can think of HLA as the display window of the cell.
The display window?
Yeah. A healthy cell uses HLA to hold up tiny pieces of its internal proteins to the outside world, allowing any passing T cells to inspect them. If a cell is cancerous, it displays mutated proteins in that window and the T cell kills it.
What?
But by shutting down HLA, the tumor boards up the display window. From the outside, the cancer cell looks completely normal, completely invisible.
The combination is just lethal then. You have no CXCL10 flares to attract the T cells and no HLA display windows for the T cells to inspect if they do somehow happen to arrive.
Exactly.
The tumor effectively throws an invisibility cloak over itself. It becomes what oncologists call a cold tumor.
But it goes even further than just invisibility. The researchers utilized single-cell RNA sequencing to look at the exact makeup of the immune cells surrounding these mutant KRAS tumors, an area we call the tumor microenvironment or TIME.
Right, the TIME.
And they found it was completely devoid of cancer-killing immune cells, but it's absolutely packed with macrophages.
Macrophages. Those are the immune cells that typically swallow up cellular debris and pathogens, right? But they can be polarized, meaning they adopt different personalities depending on the chemical signals around them.
Spot on. And the environment created by mutant KRAS forces these macrophages to polarize into an M2 state.
Okay, wait. I actually have a visual mnemonic for this that might help the listener remember.
Well, let's hear it.
When you think of the tumor microenvironment, you have M1 and M2 macrophages. Think of M2 as mute2. They mute the immune system. They're anti-inflammatory.
That's a great way to remember it. M2 macrophages are typically involved in tissue repair and wound healing. They secrete anti-inflammatory cytokines and build fibrotic tissue. Tumors often act like wounds that never heal, so they hijack these M2 macrophages to build a dense, suppressive physical and chemical wall around the cancer.
Right, so they are actively suppressing any immune response in the area. What we actually want to see in a tumor microenvironment are M1 macrophages, and the mnemonic for M1 is make one big attack, because they are pro-inflammatory.
One big attack, exactly. M1s break down that fibrotic wall and secrete signals that recruit and assist T cells in attacking the cancer. But the single-cell sequencing showed the mutant KRAS environment had almost no M1 macrophages, just a massive flood of those suppressive M2s.
So you can see why the clinical therapies were completely failing. Let's do another quick recap. You have a patient taking a PD-1 inhibitor that's designed to unleash T cells. But mutant KRAS upregulates MYC, which kills the interferon response. That stops the T cells from finding the tumor because the flares are gone, and it fills the area with those suppressive M2 macrophages. So my question to you listening is this. We have an undruggable, invisible, incredibly fast-growing tumor. How do we break the shield?
That is exactly the impossible barrier the researchers were facing. They knew a traditional synthetic drug wasn't working, and developing a new one from scratch takes over a decade of trials and billions of dollars.
Yeah.
So they turned to a massive library of 225 natural active compounds derived from traditional Chinese medicine. And they ran these really complex molecular docking simulations, basically computer models, testing how these natural molecules might physically interact with that greased bowling ball of a KRAS protein.
And out of those 225 compounds, the simulation flagged a winner. And here's where it gets really interesting. The winner was dihydroartemisinin, or DHA.
DHA, yes.
And DHA isn't some experimental untested chemical. It's a derivative of artemisinin, which is a highly effective, clinically approved, and globally utilized anti-malaria drug.
It's been used for a long time.
Right. It's affordable, and its safety profile in humans is already incredibly well documented. But obviously, an anti-malaria drug fighting cancer requires a completely different mechanism of action.
It does, and the mechanism here is mind-blowing.
Yeah.
DHA doesn't act like a traditional kinase inhibitor. It doesn't just stick to the KRAS protein and try to block it.
Okay, so what does it do?
Instead, DHA acts as a molecular tag that triggers the cell's own internal waste disposal system, a process called the autophagy-lysosome pathway.
Autophagy, which literally translates to self-eating, right? That's how a cell cleans house.
Exactly. When a cell needs to clear out damaged or unwanted proteins, it builds a membrane called a phagophore around the target, enclosing it in a little vesicle.
Like bagging up the trash.
Right. That vesicle then fuses with a lysosome, which is essentially a tiny cellular stomach filled with highly acidic hydrolase enzymes. The enzymes just digest the protein back down into basic amino acids.
Oh, wow. So instead of just trying to turn the broken switch off, DHA grabs the KRAS protein, drags it to the lysosome, and forces the cell to throw it in the garbage disposal and digest it completely.
Well, that's exactly what happens.
But wait, that brings us right back to the original problem. If mutant KRAS is a greased bowling ball, how is DHA gripping it tightly enough to drag it to the cellular stomach? Like, how does it hold on?
The researchers investigated the physical binding using a technique called surface plasmon resonance. It's this highly sensitive optical technique that measures the real-time interaction and binding kinetics between molecules.
Okay.
And they discovered that DHA is capable of forming exceptionally strong hydrogen bonds with one microscopic location on the KRAS protein, a specific amino acid residue known as serine 145, or SO145.
So it found the one tiny microscopic scratch on the surface of the bowling ball.
Yes, exactly.
And this is a huge detail. Every normal, healthy cell in your body also relies on KRAS to regulate growth, right?
Yes, wild-type KRAS.
And that wild-type KRAS also has a serine 145 amino acid. So if DHA tags all KRAS for destruction, it would be highly toxic to the patient. It would digest the healthy proteins, too.
You'd think so, but the elegance of this mechanism is in the protein folding. Even though wild-type KRAS has the SIR145 amino acid, the three-dimensional conformation, the physical shape of the healthy protein, slightly obscures that specific region.
Oh, fascinating.
Yeah, the DHA molecule simply can't reach it. It just bounces off.
But the mutations, the G12D, G12V, and G13D, they alter the physical shape of the protein just enough to expose that SIR145 pocket.
Exactly. The mutation creates a unique vulnerability. DHA exclusively binds to the mutated versions. And the researchers actually verified this incredible safety profile utilizing patient-derived colorectal cancer organoids.
Right, and for those unfamiliar, an organoid is a massive step up from just testing drugs on flat layers of cells in a petri dish. They actually take real biopsy tissue from a patient and culture it in a 3D matrix. So it allows the cells to form a microscopic, three-dimensional mini-tumor that mimics the actual complex architecture of the real thing.
It's much more accurate to real human biology. And when the researchers applied DHA to organoids containing healthy wild-type KRAS, the cells were entirely unaffected. The DHA completely ignored them.
Amazing.
But when they applied it to organoids harboring the mutant KRAS, the KRAS protein levels plummeted as they were digested by the lysosomes, and the cancer cells underwent apoptosis, or programmed cell death.
I mean, it is the holy grail of oncology, right?
Yeah.
A compound that aggressively targets the cancer while leaving healthy tissue completely alone.
It really is.
So let's do our third mini recap here. DHA, this old malaria drug, binds specifically to the SIR-145 residue of mutant KRAS and forces the cell to digest the protein, leaving healthy cells alone. But knowing all of this, a very obvious question arises for the listener.
Yeah.
If DHA is this highly specific, highly lethal mutant KRAS killer, do we even need the other two drugs anymore? Why not just treat the patient with DHA by itself and throw away the Regorafenib and the PD-1 immunotherapy?
Because cancer is fundamentally a disease of evolutionary adaptation. If you attack it from only one angle, it will almost always find a workaround. DHA is incredible, but it acts as the ultimate synergist. It's the teammate that remodels the microenvironment so the other two drugs can actually work.
Okay, let's break down that synergy, starting with Regorafenib. We established earlier that Regorafenib normalizes the blood vessels and inhibits kinases, but the paper outlines a major flaw in how tumors respond to it.
Yeah, like many targeted kinase inhibitors, Regorafenib induces significant cellular stress on the tumor. And in response to that stress, the cancer basically initiates a backup survival mechanism.
Right.
It heavily upregulates an entirely different signaling network called the ERBB pathway. This pathway involves a family of receptor tyrosine kinases that can totally bypass the blockade Regorafenib just created.
So you shut down the main power grid, and the cancer instantly fires up a backup generator to keep the growth signals flowing.
Exactly. But this is where the synergy of the combination therapy shines. The researchers demonstrated that by destroying mutant KRAS and cutting off those primary growth pathways, DHA simultaneously suppresses the activation of the ERBB pathway.
So it unplugs the backup generator.
It does. It completely closes off the cancer's escape route, which makes the Regorafenib exponentially more effective.
That perfectly covers the targeted therapy side. But the most dramatic shift really happens with the immune system, doesn't it?
Oh, absolutely.
Because when DHA forces the lysosome to digest mutant KRAS, the downstream pERK12 and PX signals finally go dark.
And when those signals go dark, the MYC protein levels completely collapse.
The silencer is removed, which means the cellular 9/11 system, the interferon response, is suddenly back online.
The transformation of the tumor microenvironment is immediate and profound. With the IFN response active again, the tumor cells resume pumping out CXCL10.
The chemical breadcrumb trails are back, broadcasting the tumor's location to the immune system.
Yes, and the cancer cells are forced to rebuild the HLA display windows, exposing their mutated proteins to the outside world. The invisibility cloak drops entirely.
So now the PD-1 immunotherapy drug can finally execute its function. And I know the researchers mapped this newly remodeled environment using that same single-cell RNA sequencing technique, comparing it to the cold tumor from before.
They did, and the contrast is night and day.
Yeah. Looking at their sequencing data, it turns into a biological war zone in the best possible way. Drawn by the CXCL10, CD8 plus T cells just flood into the tumor, a process called chemotaxis. And the sequencing reveals these T cells are highly activated. They're armed with potent cytotoxic weapons, specifically granzyme B and IFN gamma.
Granzyme is particularly lethal and is a serine protease that T cells inject directly into the target cancer cell, and it immediately triggers a cascade that forces the cell to undergo rapid apoptosis.
They're essentially melting the cancer cells from the inside out. But what happens to that suppressive wall of macrophages, the MUT2 cells?
The macrophage repolarization is perhaps the most striking finding in all the single-cell data. The suppressive M2 macrophages, which are identifiable by a marker called CD206, virtually vanish.
Wow.
The environment becomes completely dominated by M1 macrophages, marked by CXCL9. The researchers quantified this as a highly elevated CS ratio, so the entire microenvironment shifted from building a protective wall to actively dismantling it and supporting the T-cell assault.
And to prove this wasn't just working in, isolated organoids or computer models, they took this triple combination, Regorafenib, anti-PD-1, and DHA, and tested it in vivo using mice with established colorectal cancer liver metastases.
And the therapeutic efficacy was just massive. The triple combination induced synergistic tumor shrinkage, decimating the liver metastases to a degree far beyond what any single agent or dual combination could ever achieve on its own.
And equally critical was the safety data, right? Because of DHA's strict structural requirement for the mutant KRAS conformation, the off-target effects were practically non-existent.
Right.
The mice maintained normal body weight, and histological examinations showed absolutely zero unacceptable toxicity to the healthy liver tissue, the kidneys, or anything else.
And that safety profile is really what elevates this from an interesting laboratory observation to a potentially practice-changing clinical insight.
Exactly. So for our final mini recap, DHA disables the cancer's escape routes and restores the immune system's alarm bells. This allows Regorafenib to cut off the blood supply and PD-1 to unleash a massive T-cell and M1 macrophage attack. So the question we're left with is, how will this change the future of oncology? Taking a step back to look at the sheer scale of this, we are talking about a mutation that affects half of all colorectal cancer patients.
It's a huge population.
Yeah, and historically, a KRAS mutation, especially with liver metastasis, has meant a profoundly grim prognosis. The tumors were essentially ghosts invisible to the immune system and coated in a protein structure just too smooth for our best drugs to grip.
And typically, when a novel vulnerability like the SIR145 binding pocket is discovered, the clock starts on a massive pharmaceutical undertaking. You are looking at years of chemical synthesis, followed by phase 1 safety trials, just to prove the new molecule won't harm humans before you can even begin testing its efficacy against the cancer itself.
But DHA completely bypasses that entire timeline. It is already an approved, synthesized, widely distributed drug.
Exactly.
The safety profile in humans is heavily documented from its use in treating malaria. So the logistical pathway to repurposing this drug and moving it into clinical trials for cancer patients is drastically shorter. It represents a potential lifeline that could be deployed in a fraction of the usual time.
It forces a necessary reevaluation of how we even approach drug discovery. I mean, it bridges the gap between traditional botanical medicine and the absolute cutting edge of single-cell genomic sequencing and targeted immunotherapy. We mapped an incredibly complex web of immune suppression and cellular signaling, only to find the key to dismantling it was already synthesized and sitting on a pharmacy shelf.
It is a brilliant reminder that we can never stop questioning our assumptions in medicine, especially when it comes to things we confidently label undruggable.
The natural world has spent millions of years running its own chemical trials. The molecular structures it has produced often hold solutions to problems we are only just beginning to understand at a genetic level.
And that is the exact thought I want to leave you with today. As you go about your week, mull this over. If an ancient natural compound derived from a traditional malaria treatment holds the precise molecular architecture to hack a cancer cell's own garbage disposal, strip away its invisibility cloak, and dismantle one of the most notoriously stubborn genetic mutations in oncology, yet other ancient, ordinary compounds are sitting in our pharmacies right now, holding the answers to our most complex diseases, just waiting for us to ask the right questions. Thank you for joining us on this deep dive into the microscopic battlegrounds of the human body. Keep wondering, keep exploring the incredible mechanics of the world around you, and we'll catch you on the next deep dive.